I think it's my mom. Thanks for coming, everybody. There's one committee member to go. I see some people, Caitlin and Nina, Andrew. Thanks for coming. Oh, Xiaorong. Dr. Lee is in attendance. And my family, Patty's, Beth. Oh, Josh. Thanks for coming, Josh. Still waiting for a committee member. Please hold. Into, I bet you'd have time if you want to grab some food.
I just text off.
Yeah, maybe I'll stand in the foyer.
I brought the last
Super. Name. Thank you all for coming, and it's really a great pleasure, a really proud moment to introduce Erik Nordquist. He has been in my lab since 2018, 19. So he is really one of the model graduate students in our program, and has been really a role model and leader in my lab. So Erik obtained his bachelor's degree in chemistry and physics from Idaho College in 2018. And he joined our program originally thinking that he would study material science, trying to solve our energy crisis. So I'm really glad that I convinced him to join my lab and trying to solve health problem, right? Or address problems in fundamental biology and potentially can help us address some of our medical questions of the society. So it has been really a great journey, and Erik has done a lot of things in my lab. In terms of research, he has been working on fundamental approach to study the energetics of related to the regulation of different type of proteins. He has also been working on methods that integrate computational data and experimental data to construct models that allow us to predict the function of proteins in a more kind of systematic and more efficient way. And this includes some of the collaborative projects that work with Professor Lila here on some of the chaperone proteins. And he actually also work on actually several other other interventions, other folks in the department over the years. And he has also been one of our most productive graduate students. And so he has now 4 first author papers to his name. And he is working on 2 additional papers where he will be either first author or co-first author. And it's really exciting. And so I'm really glad that you can be all here to hear a little bit about some of the wonderful research that Erik has done. It's a really 5 really short years. When you blink your eye, you are here, and in a week you will be married to be a postdoc fellow in Alex McLeod's lab. Alex McLeod is one of the leaders in the field that has been working on the phosphate. One of the key developers behind the Chen phosphate. So, Erik, you have done all the hard work to earn this chemistry laser pointer.
Thank you.
On behalf of the CBI, Lynmarie, she unfortunately cannot be here. She is on remotely. So on behalf of the CBI Chemistry Biology Interface Program, I want to present to you this award of recognition of distinction for completing the CBI program. So you should hold it. So I should mention that Erik, besides all the research accomplishments, he's really one of the more celebrated graduate students in our department. He has won the prestigious Terry Endowment Fellowship, and they give a talk in the annual research fest in chemistry. So usually we'll select 4 or 5 of our best graduate students each year to showcase the kind of things they have done during their time here. While welcoming our new graduate students. So Erik was a speaker last year, and Erik also always has an inspiration to become a faculty member at the PUI, primary undergraduate institution. And so he has been devoting a lot of his time to training in terms of mentoring the fellow students in our department, in my lab, and also to do some of the teaching activities. So last year he was selected as one of the CNS
kind of teaching
fellow to offer the so-called first year seminar. Essentially, he designed the topic, the design, the curriculum, and teach a group of first year students, undergrad students, to kind of show them what chemistry can do, I guess. So it's really a special moment, Erik. So anyway, but now the floor is yours.
Thanks. Yes, and thanks everyone for coming. I want to kind of continue the acknowledgements by saying thanks to everybody in the Jianhan Chen lab, especially my advisor, who gave me such constant support. I'm trying to get rid of this thing.
You can click and hide for the music control.
Okay. Thank you. And I also want to thank the CBI and the NIH for the funding, and for my committee members who are here, Dr. Scott Auerbach, Dr. Markos Katsoulakis, and Dr. Lynmarie Thompson, who's here on Zoom, as well as all of my collaborators, some of whom are here, Lila and Eugenia. Yeah, I feel very grateful to have worked with all of you and tried to learn from all of you. And I also want to thank, I've lost it again. All of the fun memories I've had with everybody, including my my cohort who just took a photo almost all together just a few days ago. And last but certainly not least, I really want to thank my family who, yeah, really encourages me every day, especially my wife, Zoe, who is my constant support. Very grateful. So let's begin. The overview of the talk is going to follow this kind of two-pronged approach. I'll give an introduction in particular.
Can anyone hear me? We can't see the screen on the Zoom.
Okay,
Sorry.
let's address that.
Yeah, I won't see a bar.
Did I turn it off when I clicked whatever I clicked?
It's been the same the whole time I've been on, just
Okay.
a bar across the middle.
Works.
And
Great.
still see it?
Perfect. Thank you.
We'll hide the control panel again.
Yeah. Okay. Yeah, so it's going to follow this kind of two-pronged, I don't know, approach, I guess. So the first section will be about these free energy calculations of pore de-wetting, kind of 3 sub-chapters to that. I'll develop a kind of method using protein-like model nanopores to study this method of simulation, and then I'll use that method to study the effect of pore lighting mutations on the big potassium channel. And then I'll study the effect of a small molecule on that same channel. And then finally, in the last kind of section, I'll discuss the development of a predictive model of the effects of single mutations on this same potassium channel. So the broad flavor of my work has been to explore the effects of mutations or other functional perturbations on the structure and dynamics of these big molecules called proteins, the effects of those mutations on structure and dynamics, and then to try to query how those structural and dynamic and energetic changes appear as functional changes at the cellular level. So these proteins are large molecules. They're the molecules that are encoded for in our DNA. So for example, when you make some mutation to the genetic code, that shows up as a change in the sequence of this protein in its chemical composition. And then that changes how this molecule moves around and the kinds of interactions that it experiences with itself or with its environment. And that can affect the cellular function that we see. For example, this ion channel affects things like the action potential, which you can measure in the hospital through an EKG. And that's kind of what this is a picture of. And we'd like to query, how do changes at the structural level impact dynamics and interactions and appear at the cellular level as changes in function? So the protein that I've focused primarily on is a part of the symphony of ion channels that give rise to this functional output, the heartbeat. So it's just kind of one note in the overall symphony. It's called the BK or big potassium channel. It's present in many tissues, including the cardiac and the skeletal muscles, as well as in our ears. And so diseases affecting this channel are implicated in heart diseases like hypertension, neurological diseases, including autism and epilepsy, hearing loss, smooth muscle issues like bladder control, and in cancer. The structure of this protein is a homotetramer, so it has 4 copies of itself interacting. I'm just showing in this picture sort of 2 opposing copies of itself, 2 opposing monomers. Each monomer has 3 primary domains. The red here is responsible for sensing membrane voltage and then communicating that information to the pore, which houses both the selectivity filter for potassium ions. Its role is to move potassium ions from inside the cell across the cellular membrane to outside the cell. But it also houses the gate, which is responsible for kind of like a door opening and closing the channel. And the last component is this cytosolic tail domain, which is responsible for sensing primarily intracellular calcium, as well as other ligands. Both calcium binding and increasing membrane voltage can open the gate independently of one another. And that's what this figure is showing. So you can see that at 0 calcium, the normalized conductance starts in the very negative membrane voltage at essentially 0. So the channel is not conductive. As you increase voltage, it becomes conductive. The same is sort of true if you increase calcium at a given voltage, the average conductance is higher. I want to briefly introduce why this is a molecule. It doesn't look like a molecule maybe. The reason is I'm kind of representing the primary secondary structure of the protein by hiding a bunch of side chains and only highlighting the backbone of the molecule, and then representing coils that you can maybe pick out on this slide as this kind of cartoon coil, or helices, I mean. And then the loops connecting the coils or helices are represented in this kind of teal. So this is what I mean by sort of a molecule as this sort of cartoon. So the big potassium channel has a kind of mystery to it. The mystery, at least as of a few years ago, was where is the gate in this pore gate domain? So here I'm showing 2 of these pore domains. I'm blowing up just the inner helix and the filter. And I'm showing you in periwinkle the open structure, the open conformation. So the door is wide open. This is a conductive channel. And in silver, I'm showing the non-conductive closed door, except that the door doesn't appear very closed. If we compare to what would presumably be a kind of homologous structure, KCSA, you can see that the helices seem to kind of pinch off down here at the membrane interface, which would be here or here. But in BK, that doesn't appear to be the case. So people kind of were scratching their heads. Yeah, I felt like I needed to introduce molecular dynamics. So in order to kind of address this question, how does the pore open? People use molecular dynamic simulations. What those are is a representation of the universe at the atomic level. So what we do is we have a set of atoms. Here I'm just showing 2, and I can play this movie. We'll watch the atoms bounce off one another. They're kind of oscillating on this blue chemical potential. So what we do to run the simulation of 2 simple atoms is calculate the forces that the atoms experience, give them a small movement in the direction of, in the downhill direction of this energy potential surface, and then recalculate the forces, and then it kind of just iterates. And so for 2 simple atoms, it seems kind of silly, but we can construct more complicated molecular systems by adding increasing numbers of potentials to account for different kinds of molecular motion. And then we use corresponding experimental data to make sure that what we're describing is sort of experimentally reliable, or that it depicts something that is realistic to nature. And in the end, what this ends up looking like is something fairly sophisticated, more sophisticated than 2 balls bouncing. So we can describe all of the atoms in the protein, as well as a bunch of lipids, so a patch of membrane, and then all the water sort of surrounding a small box in the cell. And then we run this simulation just by calculating forces, perturbing the atoms a little bit, recalculating forces, and so on and so forth. So then if I bring back, I'm going to just zoom in on this movie here. Maybe I'll try to stop it first. Okay, it stopped. If I zoom back in and use the cartoon representation, here we can see where the gate is. The water was filling the inside of the pore, but then before I arrived in the lab, what people noticed was that water spontaneously evacuates from the inside of the pore. So here is the accounting, or we call it a trace of what happened in that movie, the black trace. So you can see that at the beginning of the movie, there were a bunch of water in the pore, and then after a certain amount of time, they all sort of left. And what's happening is called hydrophobic dewetting. In hydrophobic dewetting, the inside of a protein channel like this can spontaneously evaporate some water. And this region is kind of like a vapor lock. So potassium ions will come to this region, but they would need to desolvate in order to enter, which would incur a huge energetic cost. So either they resolvate this region and permeate through, or they remain outside. So there's no physical barrier, but potassium are locked from permeating just the same. I want to quickly explain how we might, or why I'm going to talk not about sort of counting and histograms, but free energy. Really quickly, the idea is that here we have 3 simulations. I can construct 3 histograms corresponding to these 3 conditions. One is the wild type channel, which just means the one that's in most of our bodies. And then there are 2 mutations. So this is 2 changes to the genetic code that encoded the structure and composition of these proteins. And one is red and it becomes de-wetted faster. And one is green, the D mutation, and it stays hydrated longer. You understand how that histogram is made. The way we construct a free energy surface is just by doing this log transform. And we scale it according to whatever energy unit we want. I'm going to use the energy unit kcal per mole. There is a problem here. And it's that if I want to compare these 3 mutations, it's kind of hard to see what is the total energy difference between the wild type in black, between a non-conductive and maybe a conductive state, and the same free energy difference for the red and the green curves, because in each of these simulations, you only got, in the green case, only hydrated, and in the red case, only dehydrated. So there's no comparison that can be made. So what we need is a way to sample the full cost of de-wetting in not only the wild type channel, which de-wets relatively easily, but also the red and the green. And the reason we don't just run the simulation longer, well, one reason is because a simulation with 600,000 atoms would cost about a week to get 100 nanoseconds. So we need a way to kind of accelerate this process. The method that I used, so now we're kind of in the first part, is called metadynamics. And the basic idea is kind of interesting. We're going to imagine our simulation is just a random walk in this dimension, the number of water in the pore. And we'll apply bias as the simulation goes on, such that we kind of fill in the space that we've already sampled in until we're able to sample across the full space. And if you imagine that my red laser is depicting where we are in this number of water space, we're going from an empty pore to a closed pore, but we've removed the barrier, so this can happen much faster. The other advantage is that if I remove all this red bias, I've got an image of the underlying free energy surface, which is exactly what we want. So I can just extract this as like a plaster image, a plaster mold, I mean. So we did this using model protein-like nanopores. First, to verify that the method works, we picked a pore which was relatively small and could de-wet on a timescale we could afford to sample. So this confirmed that the method agrees with itself, that the method agrees with an unbiased simulation. So one without using our metadynamics approach gives the same free energy curve as one that uses the enhanced sampling method. And we also confirmed that the method converges, which means it becomes sort of self-similar after a certain amount of time. It's essentially describing the accuracy and the precision of our method. I didn't mention, these pores are designed to be roughly the same dimension and hydrophobicity as the BK channel. So we're not sort of exploring some arbitrary nanopore. We're probing something that looks like the BK channel, and eventually we'll try to make the geometry look more and more similar to the real protein. What we first tried to do is just study how the effect of the size would vary the effect of de-wetting. We were able to do that and found something relatively boring, that the cost of de-wetting is just proportional to the exposed hydrophobic surface area and the surface area of the water vapor interface here. We were also able to include some salt and study the effect of increasing salt concentration on this de-wetting effect, and it agreed exactly with the expected colligative behavior. But that's kind of important because we want to be able to include ions if we're going to make a simulation of an ion channel. It seems important. We also recognized that the BK channel has a kind of tilt. And also the filter, if you imagine this is a cross-section of either the nanopore or of the BK channel, the filter represents an essentially impermeable pathway for water. De-wetting, if it proceeds sort of down in this way. So what we decided to do was add a tilt to our nanopores and then a lid, either on the sort of wide end or the narrow end. We thought that this would introduce some interesting complexity to de-wetting behavior, but what we found is that we could predict exactly the free energy cost of de-wetting using just the surface area based equation we derived in the last slide. So it wasn't the most exciting thing. We also introduced some amphipathic co-solvents like ethanol and glycerol, and we did see some kind of interesting behavior. So the amphipathic molecule wants to coat the nanopore with the apolar end sort of pointing towards the wall, and the alcohols pointing in where the water wants to be. We were able to sample this complex behavior just biasing the water. We didn't have to introduce a separate bias for the co-solvents. So all of this methodology seems to be working well. Let's turn it loose on the real system of interest, the BK channel. Quick reminder, it has this funny gate and it seems not to close, but we know that if you make mutations to the channel, it has a sensitive impact. By the way, I didn't mention, here's where that residue sits, right in the middle of this pore. If you make a mutation to that position on the pore lining, on the pore lining part, yeah, it's lining the pore. This polar mutation, D, makes the pore more hydrated, and this apolar mutation makes it dehydrate faster. So we employed that same methodology, but it didn't work very well, which was kind of unfortunate. The reason we think it didn't work well is because, as you can see, initially, I'm going to maybe quickly see if I can pause whoever, or maybe whoever hasn't muted, could you mute? The reason we don't think it works very well is you can see that the system is progressing through de-wetting and hydrating transitions fairly rapidly, and then it gets stuck. And the problem is that that means that the free energy bias sort of image that we're accumulating is also getting kind of stuck. All right, excuse me, it doesn't get stuck. It continues to build up bias while the system is sitting here, and then it builds up bias while it sits here. And what that results in is a kind of oscillation in our estimate. It takes like 400 or 500 nanoseconds to get a relatively converged estimate. That's kind of frustrating. So we employed something a little less sexy. It's called umbrella sampling. All it is a series of independent simulations that are just sampling one part of the region. You can think of this as like trying to climb like a rock climbing wall, but you have a series of lock-in points where you clip in. So you only need to climb 1 meter at a time, because after you get to the next meter, you clip in. So the farthest you can fall is 1 meter. So that's what we're doing here. We're clipping in at different windows and climbing the whole wall. Another detail about this is we don't have to wait sequentially. There's 10 copies of me climbing this wall all at once. Then we stitch all of that, all these histograms together into the same free energy surface we would have gotten with metadynamics. And so now let's ask what would happen if we make the mutation and perform this same umbrella sampling. And here it is. We can perform it both on the open conformation of the pore as well as the closed conformation. First observation is that the open pore remains hydrated regardless of what mutation we make. The only difference that you see here is resulting from a change in accessible volume because some of these mutations take up more space than others. The closed pore, however, there's pretty substantial changes, and you can probably follow if you know this nomenclature. Green means more polar, and this guy looks very hydrated. It's very hard to dehydrate. Whereas the wild type and the I mutation, which is apolar, they stay fairly dehydrated. They don't want to hydrate. So in order to corroborate this with experiments, we can compare to the full free energy cost of the full free energy change in opening the channel. The change in free energy, the free energy difference in opening the channel, going from the wild type or whatever to the mutant. Boy, messed that up. So anyway, that's what this delta V is measuring. And we can plot that delta V against the delta G hydration. So that's what we've just spent a whole bunch of time calculating. I'm going to define this point, about 45, as hydrated, and here, about 5, as dehydrated. And the correlation essentially means that what we've captured is proportional to the total free energy cost. So the cost of hydrating the pore is the kind of thermodynamically key step to opening the whole channel, which is interesting because we also know the channel becomes conductive when the pore is hydrated, and it's non-conductive, as I mentioned, when it's dehydrated. Okay, the last chapter in this A section has to do with this small molecule. People found that a class of molecules that look relatively similar, meaning that they have a negative charge, a hydrophobic region, and an aromatic region, all kind of in this approximate geometric orientation, seem to all act on a similar class of potassium channels. And they propose that they all act in the same way. What they found is that one of them binds inside this pore, the pore of this channel, and it seems to open up the filter a little bit, such that the channel becomes more conductive at the most conductive state. So its maximum conductance has increased. They propose that the same binding and filter opening happens in all of these channels, including the BK channel. They found this confirmation with something called molecular docking. The problem is that a subsequent study showed that, in fact, this particular molecule, one of the, let's see, it's this one, NS11021, does bind to the pore and does increase the open probability. So it does activate the channel, but it does not increase the maximum conductance. The pore is the same amount of open as before. It just spends more time in the open state. It's about 62 times more likely to be open at this very negative voltage. This must result from opening the pore. And when I say opening, I mean making it conductive. We think that because the voltage sensor domain is fully arrested. It's in the resting conformation at this very negative voltage. So what we hypothesize is that the drug is also acting on hydrating the pore. It doesn't cause the filter to squeeze open because the maximum conductance is the same, and it doesn't, yeah, I've made that argument. There's a sort of complication computationally, which is that all of these bonds, especially these two, are rotatable. And so the molecule is very, very floppy. It can access many conformations in solution. And that means that if we want to simulate its effect inside the pore, we need to wait a long time for it to tumble around and sample many conformations. And this is going to be kind of a headache. So I avoided just running a single simulation and used umbrella sampling again, same idea, except I used kind of two parallel runs. One with the charge facing down at the beginning of each bin, and one with the charge facing up towards the filter at the beginning. And then I run each simulation long enough to let the molecule kind of tumble around. Once again, each bin represents some amount into the pore, increasing as you go up into the pore. So this is sort of sampling outside, and this is sampling really, really close. I can stitch all of these together, but now we need to look at a two-dimensional free energy surface rather than a one-dimensional surface. This is the distance between the center of mass of NS11 and the filter. And this is the orientation. So positive delta Z means the charge is pointing up towards the filter, and negative means it's pointing away. Right away, what we can see is that outside it has no preference, and it's kind of unhappy relative to being inside. That's because it's kind of hydrophobic. So it immediately wants to stick. And we can kind of see that once it gets close, it prefers to be over here rather than over here. That's because in this orientation, the charge is sort of snorkeling down outside in the water, and the hydrophobic end is pointing up into the hydrophobic pore, which hasn't been hydrated yet. But once it works its way inside of this somewhat cramped pore, it can kind of point in any direction at once, except this region, which corresponds to just lying flat in the most constricted part. So everything else, it can point towards. From this binding affinity curve, we can extract something like EC50. It's actually KD, but those might be approximately the same. And what we found is that the EC50 from 2 separate experiments give a range that's almost identical to what we calculate from this binding simulation. So that kind of provides an experimental argument that what we're simulating here is relatively reasonable. It captures the appropriate physics. I'm plotting now a different 2-dimensional free energy surface. I've swapped out the bottom for the number of water in the pore. And what we can see is that what we hypothesized seemed to be true. Once the molecule enters the pore, it indeed hydrates the pore. So the last step is to figure out how does the conformation of this molecule affect the hydration? Because we can imagine maybe some conformations will stabilize the hydration and therefore potassium permeation different from others. The molecule might be blocking the potassium in some orientations and in not others. So what we decided was to select 3 independent conformations to represent our binding poses, and then sample potassium permeation independently in all 3 of these. If binding is indeed, I have it right here. If binding is indeed non-specific, so that's in contrast to the people who I showed at the first slide of this section, who believe it binds in a specific point. If binding is non-specific, then all 3 PMFs, all 3 potassium PMFs ought to agree with one another to a reasonable degree. It means that it doesn't matter how the drug enters, it just needs to hydrate, and that decreases the barrier for permeation. And indeed, that's what we found. In all 3 of the conformations I showed you, the total decrease in the barrier for potassium permeation, which is the corresponding measure to maximum conductance, is decreased by not exactly 2.5, but fairly close, about 3 to 4 kcal per mole. So this is kind of interesting because it really argues for the first time, we've found a molecule that interacts with the pore and activates the pore through hydrophobic dewetting, which is kind of a novel mechanism. And it's interesting because the BK channel is such an important and relevant, therapeutic target. This is cool, okay? I was happy. So the last section. We're going to try to extend that work to develop a model of the delta V for all mutations, which is going to be a little bit harder. So first, I want to say that the time is kind of ripe for this kind of an approach because machine learning has brought many important breakthroughs across many fields of chemistry and molecular biology, most notably in protein structure prediction. But not all problems are exactly perfect for a purely data-driven approach. So we have, in this case, a transmembrane protein of pretty large size with many interacting domains. We have multiple states, and we have interactions with several components in the cell, like the membrane and voltage and calcium binding and magnesium binding. We also have the problem that for a transmembrane protein, we're always going to be short on data because protein expression is pretty difficult, but transmembrane protein expression is very difficult. And then we're going to ask, what is the functional impact of a mutation? And those often make expression harder. sort of by biological design. So we have this trade-off between the kind of data that we can afford easily, but is low in accuracy, and the kind of data that we want, but we don't have enough of. We can solve this problem using physical modeling, thanks in part to key breakthroughs in molecular dynamics simulations, which have enabled those super fast simulations I was just talking about. We can afford hundreds of nanoseconds of systems in the hundreds of thousands of atoms. We can also afford to do more rapid modeling of the effects of local mutations on the structure, interactions, and energetics using empirical force fields that have increased in accuracy greatly over the last few years. So the goal will be to describe the physical effects of mutations on dynamics, packing, solvation, interactions, and then correlate those to experimental data that's available, which we have a fairly enormous number of mutations describing the functional impacts of those mutations on the delta V half. About 500 of them were available in the literature, but that only represents about 2% coverage of the whole sequence space of the channel. So we have a lot of data, but we need to fill in the gaps with physical modeling. So here are the kinds of descriptors that I used. I'll go into more detail on these slides. We used molecular dynamics to study the behavior, the motion of different residues in the protein. So in particular, we looked at the relative coupling between all residues in the channel and the pore, because we thought if a residue is highly coupled to the pore, it might be more or less important than a residue which is sort of decoupled from the pore, has no coupled motion. We also studied the root mean square fluctuation to sort of get at how floppy is this residue or this site, as opposed to something that's tightly packed, and how solvent accessible is this? Is it lining the pore? Is it pointing out into the free space in the cytosol? Or is it, again, tightly packed, somewhere important coupling between the voltage sensor and the pore domain? We also used those empirical force fields I briefly mentioned to describe the structural impacts, the cost to repack given mutation at both the open channel and the closed channel. The impact of those mutations on the packing, the electrostatic interactions, the cost to desolvate or solvate that residue, the impact on hydrogen bonding networks. All of this is included in this kind of quick empirical force energy function used to calculate the effect of the cost of making this mutation on both the open and the closed channel. I input all of that into a machine learning model. I used the random forest model, and I was able to extract relatively good correlation on test data. So this was fairly encouraging, and we noticed that the most important features in this final model were those, or several of them were those which we derived from physics-based modeling and simulation, especially the correlated motion between all residues and the pore, as well as the impact of solvation and the attractive packing interaction. I also want to mention that I don't know, actually, I want to talk about this. It's not that important. The key punchline here is that we trained a model without the physics-based descriptors. And the performance was significantly worse, which suggests that this physics-based modeling was sort of the key contributor to however good you would call this. But just looking at correlation isn't enough. What we wanted to do was map our predicted delta V half onto the structure and compare it between experiment and our predictions. And there are a couple of key trends. I'm going to move a little bit so I can see better. So the first trend we noticed is that the pore lights up the most. And that's important because we know, as I've just spoke for however long that was, about how important the pore is to opening and closing the channel. We know that you make a change here, it impacts everything really sensitively. So it's kind of non-trivial that as you move away from the pore, the signal decreases quickly. We also noticed that the next most darkly highlighted regions are those coupling between the voltage sensor and the pore, and the cytosolic tail domain and the pore, which also kind of agrees with the experimental data. But we noticed something a little more interesting, which is that if we take a look at this experimental data, we notice that there are several mutations of, in this case, it's asparagine, to pore lining residues. It moves the delta V half to negative, meaning that the channel becomes easier to open. I already explained why that is, which kind of takes away the excitement of this observation that the model predicts, or maybe you would call extrapolates, to all pore lining residues. Any mutation, and mutate any of these
I'm ready.
to asparagine. I can hear someone. Could you, whoever's in Zoom, mute yourself, please? Okay, thanks, Jianhan. So the reason for that would be a lot more exciting if I hadn't just spoke for however long about hydrophobic dewetting, but that is what the model is understanding, which is exciting because we didn't input any of the delicate physics about the liquid vapor equilibrium in this model. Those simulations are really expensive, so I could only afford to do it on 4 or 5. We did not do that kind of free energy calculation for every mutation, yet the model is giving it to us somewhat for free. So that's pretty impressive. But we noticed another trend. And we submitted this one to our experimental collaborators for sort of independent validation. The trend we noticed is right here at the S5 helix, which is the one that neighbors with the voltage sensor domain here and the pore here. Any mutation to L325, which is in red, shifts the V half to the right. So it makes the channel harder to open. And almost any mutation to V236 does the opposite, which is kind of interesting. And that stuck out as unusual for almost all of the predictions, even those lining the S5. We thought it was enough of a signal to submit to our experimental collaborators. I think I've already put a fine enough point on this, but we're looking to fill in the table to these 2 points. But what we noticed, I need to move again. What we noticed is that all the data we had so far kind of agrees with that trend, but it could just be a coincidence. In fact, it wasn't. So what we found was relatively great correlation between the true and predicted V half shift. Means that, yeah, if you make an H mutation to this L235 and V236, it has the opposite impact. Same for alanine, same for tryptophan. It suggests that this this S5 position plays a particularly important role in coupling between the voltage sensor and in the pore, such that if you make some change to L235, it seems to make the pore a little bit more closed, and the voltage sensor seems to want to pull it back in, something like this. It's a little hard to speculate because we would need to run a bunch of additional simulations and possibly additional experiments to really investigate this site. But the fact that the model was able to pick this out as sort of a key hotspot from among tens of thousands of possible mutations is a really encouraging result to confirm that our model is working. And more importantly, that the physics-based descriptors we inputted at the beginning were the key contributor to that success. So here at the end of 5 years, yeah, I'd like to just give a quick recap of what I've accomplished today. We use this enhanced sampling protocol to really do 2 things. One was to show that, yeah, if you make a mutation to the pore, it has the effect it has because it's changing the free energy of hydrophobic dewetting. And that the same thing is true of this drug, that it impacts the dewetting, and that's the only thing it needs to do. It just needs to be in the pore, and in any conformation, the effect on conductance is the same. And then finally, on a really challenging problem, we were able to pick out a novel hotspot coupling between the voltage sensor of this potassium channel and the pore, and showed that in at least these 4 mutation cases, the model is picking out something real and reliable. And that one ought to be able to use this kind of modeling scheme in many cases, other proteins, where data is kind of scarce and you need to use physics-based modeling to fill in the gaps and construct a predictive model like this. So here at the end, thanks everybody. I could have guessed, Jeannie is first.
Well, so even from the very first time you showed the channel, the part that stuck out to me was in that central core, one helix over from your S5 position, there's a bulge in the helix. So has anybody investigated the role of that bulge?
I want to make sure.
So right above where you have L35, one rung over,
This one,
no, the next helix toward the left.
this one.
Yeah.
Yeah, that's a good eye. So there's a lot of problems with that bulge.
Could you make sure?
You can see it on the other side better.
Right
So
there.
if you look at the, yeah.
This.
Got it.
So that is where the pore hinges. So there's a double glycine there. And I have like the open and close change. Yeah, so that's where the S6 hinges. So you picked it out. People have looked at that.
What happens if you get rid of one amino acid?
Delete it?
Delete one, because then that might pull, that should pull in the bulge, and then you'd have like a normal helix. Does the core not function?
I'm not certain. I'm not certain. Do you know that?
So that bulge actually is a little weird. I think if you look at the structure, I think it's a upper end. It doesn't it's not really a high bulge. So in humans, I think it's a.
Yeah, I didn't want to. There's other
There
problems
was
with the
like
structure. Yeah.
In this other fungi sequence, there's a glycine somewhere near there, but for human sequence, glycine is much closer to the filter. So I don't know which structure you use to plot this.
This is the human.
The crow yam original. So that structure has contain a mysterious character
Yeah.
in a closed state, but that is I don't think it's real. It's actually really unstable in our simulation, so it will go away.
But this is post relaxation, so it still persists a little bit. But this is not as I'm impressed that she noticed. It's not as very sharp.
I worked on a bulge.
Yeah, so first of all, very nice.
Thank you,
Really neat story, and really nice accomplishments that you've had. When you get to the machine learning part at the end and you're putting in all these different variables, I was wondering why you didn't put any evolutionary data, because you
I did.
talked
I'm
about,
sorry.
trying to come up with covariance and things, and I'm thinking, oh, I know where he's going. He's going to talk about multiple sequence alignment, where the variation is, how they're correlated, and all that. And you didn't go there at all.
Yeah, that's because I forgot to mention that I did include a score from I don't know, an online tool that does multi-sequence alignment and gives you a fitness score that uses that information.
I was thinking sort of the Brahman-Ganathan type approach, and I was wondering too, how many of the representatives that are out there have been characterized in a way that you know something about their potential, and whether there's any correlation that can be used from that.
What I know is that we used all of the information where the sequence matches. So I have included mouse, rat, and that fungus, but wherever the sequence matches, but I didn't look at places where it's different.
I'm interested in your interaction of small molecule channels.
Yeah.
How do we know that it was actually a non-specific binding? How is that determined? And more importantly, I wanted to get your take on a small molecule importance of small molecule dynamics in channel binding is because you're having exchange between various conformations here.
Mm.
And in drug discovery, we've been trying to include dynamics of small molecules in drug design.
Mm.
Unsuccessfully so far, but does it really matter? At least in this case.
Yeah, good questions. So we used NMR data in solution to confirm that what we have, the conformational landscape that we have in solution of this molecule agrees.
So this is without the channel.
Yes, without the channel, it's floppy. The only experimental data we have, other than we know that it binds in the pore, is the EC50. So we know that we get the same approximate binding affinity as we would expect. The only other piece of experimental data I can give you is that not in the BK channel, in a separate channel, they have a structure with the corresponding molecule for that channel, the same part of the class bound. But the only thing that's resolved is the large, there's a large halogen. So everything else is invisible, which would suggest that it's flopping around. The final, I'll wave my hands a little bit here. If we look at the pore, there's a lot of space, and it doesn't really look like a receptor. So it was always kind of strange in my mind, and I think in Jianhan's mind, that we were looking for someplace to wedge this guy into just a tube. And so finding that there was no evidence of a tight binding, that it just sort of comes in and gets stuck, to me makes a lot of sense. It would have been more weird to find that it rests somewhere. Those are my answers. I forgot the second question.
I think the most strongest evidence is that structurally they cannot see. I mean, they can resolve the rest of the channel to like 3 or 4 amps.
Yeah,
They
three.
cannot see the bound drug at all. They can only see it when they label with a heavy atom, that they see the atom automatically. So the drug is bound, but I think the most kind of logical explanation is that the binding pose is not a specific binding pose. It's either dynamic or multiple binding pose.
Right.
So, do you want to keep your question for the closed session, or you have a do whatever you want? Either way, so if it's a tough question, we'll keep it. We'll stop. Yeah, but please stop. Go ahead. It's a softball. There's no softball on Scott.
So you mentioned earlier in your talk the KCSHN, and I'm curious if there's a substantial similarity in the selectivity filter between BK and is that sort of across the board?
Yeah.
See?
I want to.
That was easy.
I can even spell, I think, TVGYS.
TVGYG.
TVGYG.
Yeah.
I can't spell.
I did not know the sequence of the field.
That's the only thing I know.
Yeah. So fairly similar.
Markos, please.
Maybe softball. So you told us this great story about trade-offs, scientific machine learning. It's a big in the area, right?
Yeah.
Scarcity of data.
Yeah.
And some physics.
Yeah.
Knowledge. So can you comment more? Did you probe the edges of this? I mean, which were the most significant physical descriptors?
Yeah.
Try to go long data and see what happens.
The I don't know, punk answer is that the random forest model doesn't need you to drop features even that are correlated. So I didn't have to worry too much about that. Maybe that was why I picked it. But we did look very carefully at the feature importance, which is that mean decrease in the impurity score. And fairly consistently, the most important features were correlated motion, fluctuation of motion, and the change in solvation which I expect has to do with the fact that those key mutations that line the pore have a big change in solvation free energy. And even the Rosetta calculation can capture that. But the dependence was fairly weak. Like I think I showed you in the data defense that there was no correlation between any of these features and the delta V, even the total Rosetta delta 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 G. So we had to kind of, yeah, not worry too carefully if any of one of these features is working.
Erik, is that time for a question? Is there time for one more question? Can you hear me?
I can. Can you wait for? And I also think
Oh, I'm sorry. Yes. Sorry, please.
We'll have the question for Alejandro first, then.
You mentioned these two mutations, right? There were two hydrophobic amino acids. When you mutated one, it goes one way. When you mutate the other, going the other way. What happens if you mutate both?
Oh, you're talking about at the end.
Yes.
That's exactly what our collaborator asked. The model can't tell you, or at least I don't trust to ask, but we didn't test that. I think that would be the next thing. One of the next things I would have tried. If I was to ask the experimentals to do something, it would be that. And if I was to do something, it would be to run a simulation and tug on these guys and see what the mechanical coupling is. That's a great question. Especially since the double alanine has the same effect as the double tryptophan. It's really weird. Tryptophan's big and alanine is small. It's a very strange dichotomy. Mattia, I am ready for your question.
Great. Sorry if I interrupted anyone. It's hard to hear the room. Yeah, great talk. Really love the work. Your presentation was clear. First of all, congratulations. So my question is about the last part of the machine learning. It was very interesting that I believe you found a mutation in the voltage sensing domain that correlates with the de-wetting. Is that correct?
It's on S5 actually.
Yeah, wonderful. So do you know so that is pretty interesting. You basically can manipulate the voltage sensing domain to open the pore or modulate the pore. Do you know if there are any potential pockets there have been reported, or do you think potentially there is a chance a small molecule or peptide can bind to that region and access from the membrane domain?
Spoken like a true pharmaceutical, industry worker. I don't know of any pockets, but there is that. Where's my cursor? Now I just realized all the Zoom people couldn't see my laser this whole time. But anyway, I can't get my mouse, but you remember that there are those water-filled crevices between the S5 and the S6. I don't really remember exactly where those sit, but it could be that one of those is accessible to V236. And then this loop is dynamic, so I would imagine you can at least wedge kind of into that, but I don't know. I don't remember exactly.
That is quite interesting. Thanks for sharing that. And again, congratulations.
Thank you.
So any questions from audience? If not, this concludes the open session of the defense. Thank you all for coming.
Yes, thanks.
We'll stop.
Oh, yeah. Thanks for coming, everybody on Zoom. I respectfully ask that you leave the Zoom so I can just talk with them. Hey, thank you. You have to go. Got an experiment ready. Thanks so much for coming. Are you coming to Wednesday? Hey, you got a lot of fun. But yeah, thank you. Just remember. See you, man.
Yes.
Thanks. Thanks. If you didn't, it's probably on me.
No, it was pretty, yes, you did a good job of making it difficult.
Thanks.
Nice.
How do you like that?
Great. I like the picture.
It's done. Well, it's almost done. 5 more minutes.
Where you come to now?
Yeah, I'm gonna take some photos. I have my robes in my car.
Oh, this. How did you get this?
My mom gave it to me when I came here.
Oh, nice. I like
Yeah.
this.
I like it too.
I
It's
even
got
get it.
chemistry
This is
stuff
the
on it.
chemistry style, yeah. I like it.
Thanks.
See you then.
Yeah, I'll be out there in a minute.
I like that.
Scott brought a dog, Lynmarie. So there's some conferring about the dog.
That's good.
Okay.
You did a great job.
Thank you, Dr. Thompson.
Take care. Bye-bye. So I ran into your daughter.